history about this genetic molecule. Now, the discovery of the DNA is quite later, about 1950s. Now look at this, the discovery of protein much more earlier, this discovery of protein. So when scientists know about the protein, we know that protein is a main, what we call substance for the metabolic reaction, particularly in terms of the enzyme. Are you clear? So when we talk about genetic molecule, scientists actually define this genetic molecule must be able to carry the instruction, to instruct the cells what to do, and they can be perfectly copied and passed down to the next generation of the cells. Because in the first chapter, we learned about the cells theory, when we talk about what? All cells must arise from the pre-existing cells. So it means that whatever instructions must be able to copy down. Okay, so at first, scientists think that it's a protein, but protein only can carry instructions. Means that something like carry instructions, the cells will do once, but it's not true. But protein cannot be copied. Protein cannot be copied. Protein is a final product. So means that until 1950, Watson and Crick, these two persons discovered the structure of DNA, and that is the start of the new branch of the study. We talk about these genetics and epigenetics. Okay, so nowadays everything we talk about the genetics, right? The disease genetic, DNA, genes, genome. Okay, so those are the genetic molecules we involved. For example, the word DNA. So why we purposely put this carry instruction and copy? Because in this topic we're going to learn two processes which are very, very important in terms of genetic study. One is DNA replication, which fall under perfectly copied. I purposely put a brackets here because none of ourselves can copy exactly. Are you clear? Ideally, copy exactly. When I ask you to copy something, sometimes you also copy wrongly, right? Okay, so ourselves, same thing. Okay, if we copy wrongly, but we do have a mechanism to stop it. Okay, so carry the instructions, basically our gene. So we're going to learn this in protein synthesis. So the first part refer to the protein synthesis. Second part refer it to the DNA replication. Okay. So the instruction carried by genetic molecule are used for the constructions, behavior of cells, and how the cells have to grow to form a complete living organism. If let's say we don't give the instruction, what will happen? Your cell won't know what to do. And you can see that us, as a multicellular organism, basically is a meatball. You can imagine no specialization, no bone, no eye, nothing. Basically a meatball. Just a very big meatball. Are you clear? Okay, so that's why the genetic play a very important. Okay, so whenever the cells divide, the ability of genetic molecule to be copied perfectly over and over again is allowed the exact copy of instructions to be passed on to the nucleides of the daughter cells, which we are going to learn about how this mitosis takes place in next chapter. Okay, the mitotic cell cycle in chapter 11. So in our syllabus, we're going to focus on these two kinds of the genetic molecule, DNA and also RNA. RNA itself, so we have many kinds of RNA. So RNA we learn here is in general. So let us look at the structure of the DNA and RNA. So DNA and RNA, we call them as nucleic acids. So from the word acids, we need to know why. They are macromolecule, as I say that, because they are many similar repeating monomer joined into a long chain, which is known as polynucleotides. Generally, DNA consists of two polynucleotides. RNA consists of one polynucleotide. Are you clear? DNA double. Right? We say they have double polynucleotides. So the repeating monomer from which DNA and RNA molecule are made is known as a nucleotide. So what is the structure of nucleotide? If you look at the nucleotides here, it's a DNA molecule. Nucleotide consists of three parts. You can see that the projecting sideway. Can you see that? This we call them as a nitrogen-containing base or nitrogenous base. And this structure, you can see that why called nitrogen-containing base? Because at this structure, you can see that they contain nitrogen. A lot of nitrogen. Can you see that? Nitrogen's atom. So that's why this part is termed as nitrogenous base. So in DNA, we have adenine, guanine, cytosines, and thymine. And two ring structures is known as purine. Single ring structure is known as pyrimidine. Can you see that? So this is the first part. Second part, pentose sugar. Why called pentose? 5 carbons. We learned about hexose. Hexose, 6 carbon. So here, pentose. So because we have the carbon number 1, carbon number 2, 3, 4, and carbon number 5. Can you see that? Carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. So this one actually is the ribose, pentose sugar. So we have 2 kinds of pentose sugar. We have the deoxyribose and ribose. So what makes them different? Look at carbon number 2. One is H, one is OH. So from the name, D means remove, oxyribose. Can you see that? The name D means remove, oxy means oxygen from ribose. So therefore, at carbon number 2 here, OH group. Can you see that? It's the OH group. At carbon number 2, OH, now oxygen atom is removed, become H. Can you see that? Again, guys, the OH here is removed, the O. I clear this ribose. This is ribose. So the name deoxyribose, D means remove, oxy means oxygen. So remove oxygens from the ribose sugar. So we get deoxyribose. So deoxyribose in DNA, ribose in RNA. Are you clear? Clear? And last part is this red color thing called phosphate. So phosphate is the one that actually gives the acidity to the DNA. So DNA, they actually charge negative. They are acidic. So that's why we call it as a nucleic acid. Are you clear? Okay, phosphate group actually come from the phosphoric acid. So they have the ability to donate protons. So as long as the items that have the ability to donate proton, they are acid. So that's why we call them as a nucleic acid. Are you clear? Clear? So now we go into these parts, the nucleotides. Just now I have briefed you guys go through these 3 components. So first we have the nitrogen containing base. We have 5 carbon pentose and a phosphate group. Okay. So this is the structure. So we have the nitrogen's base. We have the sugars, pentose sugars, and the phosphate group. So the pentose sugar 5 carbons can be either ribose or deoxyribose, depending on the hydroxyl group OH attached to carbon atom number 2. So if the ribose, then if OH group. If let's say okay, it's the deoxyribose, then hydrogen atom. So from here, you need to recognize the pentose sugar. Activity 10.1, you can see that the first A and B, so it means the pentose A and B. So pentose A, check carbon number 2, always remember check carbon number 2. So you can see that carbon number 2, H, no oxygen. So A actually is deoxyribose. Okay, A is deoxyribose. How about B? B with oxygens, right? With the oxygens there, oxygen atom there. So therefore, carbon number 2, and carbon number 2. So therefore, B molecules is termed as ribose. Okay, it's ribose. Here. So there are 5 different nitrogen containing base, ring structure with a nitrogen atom found in the DNA and RNA. So this one you have to know, highlight guys. For the DNA, ATCG, adenine, guanine, cytosines, and thymine, ATCG. But the thymines here, in DNA, we have thymines, but RNA thymines will be replaced by uracil. Okay, so DNA, ATCG. RNA, AUCG. Thymine is replaced by uracil. Okay. So adenine and guanines, they are purine, basically double ring structure. cytosine, thymines, and uracils, they are pyrimidine single ring structure. Now, so this is the way I normally use to remember. I mean, until now, it's still, I mean, it's difficult for some students to remember. Okay. CUT. Cut. So cut pyrimidine. So pyrimidine is cut, therefore only one ring. Okay, this is how I used to remember when I studied. So pyrimidines, they are single ring structure because they are cut. So it means that who are the pyrimidine? Cytosine, uracil, and thymine. How many rings? One ring. Why? They are cut. From two rings, they are cut into one ring. Are you clear? This is how I remember. But nothing much, it's just the way you can find your own way. So in this structure, you can see that, guys, we have the two-ring structure, adenines and guanines. So my mistake, guys. This is uracil. Okay, it's not guanine. It is uracil, guys. Change it. So uracil and thymine actually differ by one functional group. But do you need to remember them? The structure? The answer is write down, no need. You are not required to remember the structure of the bases, but you need to remember how many rings they have. Okay, so adenine, okay, and guanines. Adenine, guanine, two rings, they are purine, CUT, okay, cytosines, uracils, and thymines, single ring. Okay, so complete this table. Which are the purines that only appear in DNA only? Which are the pyrimidines that appear in the DNA only? Okay, try to complete this before I give the answer. Okay, so how many purines we have? Two purines, adenine and guanine. And adenine and guanine both appear in DNA and RNA. So the purines, no issues at all. Okay, DNA only, RNA only, no. So adenine and guanine. Okay, so both of them, adenine and guanine, both of them appear in RNA and DNA. Eh? These two pyrimidines in DNA only, pyrimidine in DNA only, thymine. Okay, RNA only, uracil. Okay, and cytosine is the one appear in both RNA and DNA. Okay. So how about phosphate group? Phosphate groups, they join the pentose sugar by condensation. So at this moment, I won't go into the detail about how the phosphate group actually join the nucleotides together, right, to form a long chain. But basically, the process by condensations. So therefore, polynucleotide is a macromolecule. Are you clear? By condensation process, side by side. Okay. So it gives the nucleotide acidic property because of the negatively charged, or we say that they can donate protons. Okay, they can donate protons, so therefore they are acidic. Okay, so this diagram summarizes the entire structures. Here you can see that we have pentose sugar. Pentose sugar, we have either ribose or deoxyribose. And in terms of base, you have purines and pyrimidine base. So purine base, two rings, and pyrimidines, one rings. Okay, one ring structure, and another one is the negatively charged phosphate group. So three components, right? Three components to form a nucleotide. Okay. How about polynucleotides? When you talk about polynucleotides, they are linked by the condensation process. Linked by condensation process, alternating a sugar phosphate backbone. Okay. So here we have a little bit of things to learn here. So the 3' carbons of one sugar is bonded to 5' phosphate of the adjacent sugar to form a phosphodiester linkage, and the bases projecting sideways. Okay, now don't worry about this part, because when it comes to the DNA, I will draw out the structure and show you guys what this means by phosphodiester bond. Are you clear? Okay, so we have two types of polynucleotides that we are going to learn. They are DNA and RNA. So the first thing we're going to learn today, will be DNA. Okay.